So we will discuss the placenta formation and then the functions of placenta. The first diagram you can see that the chorionic villi, the trophoblast is penetrating to the endometrium and making this chorionic villi. And later in the second diagram where we have much more focused on the chorionic villi. So inside this chorionic villi you can see that the blood vessels being developed, and then later these blood vessels which develop inside the chorionic villi will get connected to the embryo through a structure which is known as umbilical cord, which we'll discuss later. Anyway, first the trophoblast cells will penetrate to become the chorionic villi, and then blood vessels will develop inside the chorionic villi, and then the blood vessels in the chorionic villi get connected with the embryo through the umbilical cord. And this is the detailed diagram of the placenta, where you can see this is our embryo surrounded by the amniotic cavity. Amniotic fluid is there inside that, and then this part is the placenta. The entire part, you can see the entire part is actually considered as placenta, and you can see the placenta is connected to the embryo. The placenta is connected to the embryo through what? The placenta is connected to the embryo through the umbilical cord. Once again, the placenta is connected. The placenta is connected to the embryo through what? The embryo through the umbilical cord. Okay. Now on this diagram, we can see very clearly this is the placenta, and half of the placenta is from decidua basalis, which is the endometrium between the embryo and the myometrium after the implantation of the embryo, and then the remaining half of the placenta is from the chorionic villi, which develop from the trophoblast. So half of the placenta is maternal and half of the placenta is fetal. So if you see the details of this diagram, you can see as this chorionic villi is penetrating to the deeper part of the endometrium, the maternal blood vessels will break, and this blood will get filled inside certain cavities, certain spaces. And now this chorionic villi is actually embedded inside the blood of mother. Oxygen and nutrients will be released from the maternal blood that will diffuse inside the chorionic villi. From there, it will move towards the embryo through the umbilical vein and umbilical arteries. The waste materials from the embryo will be released to the pool of mother's blood. From there, it will be absorbed to the mother's body. So it's very simple. Placenta is the area where the exchange of materials are happening. From mother's body, oxygen and nutrients will deliver to the developing embryo, and from the embryo, the waste materials, carbon dioxide, etc., will be released to the placenta. From there, it will be taken to the mother's body, from where it will be excreted or released out. So, this is the structure anatomy of the placenta. And you can see there will be two umbilical arteries and one umbilical vein. Umbilical vein is in red color because this is carrying oxygenated blood towards the embryo, whereas umbilical arteries are in blue color because they are the one which is carrying the deoxygenated blood, the uh, blood which is carrying waste materials from the embryo, carried from the embryo to the placenta through these umbilical arteries, which is based on the definition of the arteries and vein. Now the functions of the placenta. Already explained that exchange of materials between the embryo and the mother, oxygen and nutrients from the mother will be exchanged to the embryo through this placenta, whereas carbon dioxide and waste materials from the embryo will be exchanged to the mother in the placenta. It is also acting as a barrier which prevents the entry of unwanted substances from the mother to the embryo. Like, unwanted pathogens, unwanted chemicals kind of things will be prevented. Even though it prevents the entry of many unwanted substances to enter the embryo, some of this, like HIV, hepatitis B, Hep B virus, rubella virus, or certain drugs like thalidomide drugs can cross the placenta and may cause problems in the development of the embryo. And this kind of substances, this kind of agents which can cross the placenta and cause problems in the development of embryo is called teratogens. So teratogens means terato means monster. Gens means generating. Monster generating substances, agents which can cross the placenta and can enter to the embryo. So we call them as teratogens. And then it is acting as a temporary endocrine structure. It releases few hormones which is important for the development of the embryo, maintenance of the embryo, maintenance of the pregnancy. One of the most important hormone is human chorionic gonadotropin, which is required to maintain this corpus luteum because LH is normally maintaining corpus luteum, but later stages after fertilization, what happens? LH level declines, but by that time, placenta will start to release HCG. Now, HCG will maintain the corpus luteum, and corpus luteum releases progesterone and estrogen, which is required to maintain the endometrium, and it is required for mammary gland development and other preparation for the mother's body. It also releases, placenta also releases relaxin. It increases the flexibility of this pubic symphysis and helps uterine cervix during the labor. It releases human chorionic somatomammatropin, which is also known as human placental lactogen, which helps the mammary gland or which prepares the mammary glands for lactation. And it also releases CRH, corticotropin releasing hormone, which establish the timing of birth because this actually increases the secretion of cortisol. And you can see at the time of parturition or nearby parturition, the CRH level is going to reach the peak. So it helps in parturition, delivery, labor, which fixes the timing of birth. So HCG, relaxin, HCS, CRH, as well as not only that, estrogen and progesterone is also being released by placenta. Okay, estrogen and progesterone also released by placenta. So this is the structure and function of the placenta.